Calcium plays a huge role in it. So your cells need it around, so they store it in the sarcoplasmic reticulum. But again, other things to see here. Go back to the plasma membrane and look back at the sarcolemma, that blue stuff you see in the picture. If you notice, it looks like some of that plasma membrane, some of that sarcolemma is kind of oozing down into the cell as these little blue tubes. We call those transverse tubules, or a T-tubule for transverse tubule. And that's just an extension of the plasma membrane. And its job is to help conduct the action potential, help conduct some electricity deeper into the cell. And we'll see that a little later on in the chapter. Turns out you're going to need to conduct some of this electricity into the cell to reach the sarcoplasmic reticulum to cause it to release calcium. That's why when you look at the picture, you always see the T-tubule kind of wedged in between neighboring sarcoplasmic reticulums. And we even name this. It forms like a cookie sandwich, an Oreo cookie sandwich, where the cookie wafers or the ends of the sarcoplasmic reticulum, we call them the terminal cisterns. A terminal cistern is just the end to a sarcoplasmic reticulum. And sandwiched in between, like the cream in your Oreo cookie, is the T-tubule. We call this a triad. I think tri for three. A triad is just one T-tubule and two neighboring terminal cisterns. And this structure is going to help to conduct the electricity from the exterior towards the interior of the cell to help reach the sarcoplasmic reticulum. Don't worry, we'll talk about it when we get into the physiology. Right now, just know the parts. A triad is one T-tubule and two terminal cisterns, two ends of two neighboring sarcoplasmic reticulums. What else? There's one more major thing you see inside of the cell. It's what's taking up most of the room. It's these long red rod-like structures in the cartoon. Those are another type of organelle in the muscle cell, and we call it the myofibril. You can see the muscle cell is filled with myofibrils, these long little rod-like structures. And these myofibrils are what we call the contractile organelle, meaning it's the organelle that's going to help you to do your muscle contraction. And by the way, to contract simply means to shorten. So when you contract a muscle, what you're really saying is you're shortening the muscle. And when you shorten the muscle, that will pull on your tendons, which will pull on your bone and help you to move. So when you talk about contractions, even with a female, when she's having uterine contractions during delivery, she's really having muscle contractions. She's shortening muscle in her uterus to help get the baby out. But all this shortening is really the result of this organelle. When the muscle or the cell contracts, when the cell shortens, it's really because the myofibrils have shortened. So we say it's the contractile organelle. It's the organelle responsible for the shortening. That's why muscle cells are crammed filled with these organelles, because muscle cells need to shorten. They need to contract, because that's really how you're going to move. So those are the basic organelles that we see. The sarcolemma is just the plasma membrane forming the outer boundary. Everything inside is called the sarcoplasm, not cytoplasm. It still has nuclei, mitochondria, but then we see other things like the sarcoplasmic reticulum storing and releasing calcium, and myofibrils helping with the contraction. And again, we're going to go in a little bit deeper. This is about muscles, and muscles work by contracting. So we're going to look in on that organelle that helps us to contract. We're going to focus in on the myofibril. And that's what you see here on this picture. You're looking inside of a muscle cell, and the long red rods are the myofibrils. And if you notice, it looks like it has some familiar characteristics. I see some alternating light and dark areas. And we name these areas. It's broken up into repeating segments. And you can see one segment highlighted on the picture. Your myofibril is just made up of repeating units we call sarcomeres. So in the image, they've highlighted one area that represents one sarcomere. And it just repeats over and over. And that entire unit is called the myofibril. So to help explain contractions, we're going to zoom in and focus on a sarcomere and kind of look at what's going on. I want you to know the anatomy, the parts to a sarcomere. Why? Because you can see it's making up the myofibril. So when we say a muscle cell contracts or a muscle contracts, we know it's really because the cell is contracting. And now you know it's really because the myofibril is contracting. We're going to take it one more step. It's really happening because the sarcomere is contracting. So we call the sarcomere the contractile unit, or the smallest contractile unit. It's really the first place you're going to see the shortening or the contraction occur. So we're going to slow down here and kind of spend some time talking about sarcomere anatomy, which is what you see on this image here. Down below, you really see what's making up a sarcomere. It's really a bunch of proteins all held together. That's why muscles really have a lot of protein in it, because the individual parts of it, or at least the major organelle, is made up of lots and lots of proteins. And we're going to focus in just on a couple of the proteins here. For example, some of these proteins help us to highlight the beginning and end to a sarcomere, basically the boundaries of a sarcomere. They're what we call the Z-discs. So on the image, you see on either side is a Z-disc. And it's called a Z-disc because when you look at it, it's this blue line you see, kind of zigzag looking line. That's why it's called a Z-disc. But it really forms the boundaries to your sarcomere. So we say a sarcomere stretches from Z-disc to Z-disc. Those are the boundaries. So you could tell where a sarcomere will start at one Z-disc and it will end at another Z-disc. But that's not really their job. That's just what they happen to do. They happen to be the boundaries. Z-discs are what we call structural proteins. They're proteins that are going to provide structural support for other proteins, meaning they're almost like holding up other proteins. That's what it means to provide structural support. And you can see who it's providing structural support to in the picture. There's these long yellow rods attached to the Z-discs. That's another protein. It has lots of names. It's called actin, and it's a type of thin filament. A filament is just a thread-like protein, and this one is extremely thin, so it's called a thin filament, and its true specific name is called actin. So actin kind of attaches itself to the Z-disc and uses the Z-disc for structural support. What else? Oh, if you notice, there's a protein running right down the middle of the sarcomere. It looks kind of teal in the picture. And it's running right down the middle, almost in a straight line. So we call this protein the M line. It's another structural protein, just embedded right in the middle of the sarcomere. So we call it the M line. And again, it's providing structural support, and you could also see who it's providing it for in the picture. You see this long, thick, red rod-like structure attached to the M line. That's yet again another protein. It's really thick compared to the thin actin filament, so it's called a thick filament. And it also has a specific name. It's called myosin. So myosin uses the M line for structural support. And really, how you contract the sarcomere, how you contract the myofibril, the cell, and the muscle is really because of the interaction between myosin and actin. It's really because of the interaction between the thick and thin filaments. So those are the major proteins you're going to see in your sarcomere. Z-discs forming the borders, providing structural support. The M line in the middle also providing structural support. And then you'll have your thin actin filament and your thick myosin filaments also. And then to finish off pointing out some sarcomere anatomy, we look at areas. We not only name structures, we name areas in anatomy. And so the next things I'm about to name are just areas, regions of the sarcomere. For example, we name the areas where you only have thin filament with no overlap of thick filament. So you can kind of think and look at the picture. Where do you only see actin and no myosin? Only see the yellow bands and no red bands. Turns out those areas are what we call the I band. The I band is just the area where you have only thin filaments with no overlap of thick. And if you look at the picture, you notice the Z discs run right down the middle of your eye band. So when we look at a sarcomere, your Z discs have cut the eye band in half. So when you look at a sarcomere, you're going to see half of an eye band on one side and half of an eye band on the other side of your sarcomere. So when we talk about a sarcomere, we say it has two halves of an eye band, one half on each side. There's another area to name. It's called the A band. The A band is basically the length of your thick filament. It's the length of myosin. So if you were to draw a line at the tip of myosin on one end and a line on the tip of myosin on the other end, everything in between is called the A band. It just runs the length of myosin. And right in the middle, kind of around the M line, there's an area where you only have thick filament with no overlap of thin. So right in the middle, right around the M line, you see there's only the red myosin filament and no actin filament. We name that area. It's called the H zone. The H zone is just the area where you have only thick filament with no overlap of the thin. So that's basic sarcomere anatomy. Z discs form the borders. It has actin, thin filaments, and myosin, thick filaments. It has the M line right down the middle. It has two halves of an I band, one A band, and one H zone. That's basic sarcomere anatomy. And when you look at the sarcomere, believe it or not, you've seen some of these parts to the sarcomere already. You've actually seen some areas of a sarcomere under the microscope. You've seen the A and I band. How? Well, you see from this table 10.1, under the microscope, when we looked at skeletal muscle tissue, remember one thing we saw were striations. We saw the alternating light and dark bands. Well, when you were looking at striations, you were actually looking at alternating I and A bands. How? Remember, the I bands are where you only have thin filaments. So when you put that under the microscope and turn on the light, it's like you having very thin blinds. So the light's going to come through it really easily, and that's going to look like a light area. And you remember the A band ran the length of myosin, which was thicker. And if you go back to the picture of the sarcomere, the A band will have some overlap of actin in there with the myosin. So it's like you have thicker blinds and extra blinds. So that's going to block a lot of the light. So the A band is going to look darker. So when we were looking at skeletal muscle tissue under the microscope in lab before the break, you were actually looking at alternating A and I bands already. You just didn't know it. And so when you look at a muscle, again, I told you muscles have lots of proteins, and it's all because of all the proteins you see within your sarcomere. And again, we've seen these two already. We've seen two of the proteins that are responsible for the contractions. That's why we call them contractile proteins or contractile filaments. They're actin and myosin. So when we talk about the physiology of how you move, we're going to really be talking about how do actin and myosin interact. And remember, this is skeletal muscle tissue. So they're only going to interact when you allow them to. So we need something to prevent them from interacting unnecessarily. We need something to help us to regulate when actin and myosin can interact. Turns out that's the job of other proteins in your muscle cells. We call them regulatory proteins. There's going to be two major ones. They're called troponin and tropomyosin. They're called regulatory proteins because they're going to help to regulate when myosin and actin can interact. How? Well, if you're not moving, well, you don't want actin and myosin to interact. And the way you're going to do that is that troponin and tropomyosin are actually going to cover actin. You could see from the picture down below, troponin in the cartoon is the little blue looking structures, and tropomyosin is the long golden rods. And to help regulate contractions, they actually sit on top of actin and block myosin binding sites, which are just sites on actin where myosin will need to bind when they interact. So to keep you from moving, troponin and tropomyosin block the myosin binding